Welcome to Science Breakthroughs from Science Mission. I'm Sadashiva Pai and I'm going to discuss a couple of breakthrough papers published in the week of May 12th. Let's start. First, we are going to discuss a paper published by Marina from Dr. Su Chun Zhang's lab in Cell Reports. The paper takes a leap in the regenerative therapies by demonstrating successful use of autologous iPSC cells in monkeys. Why this paper is so important? Teratoma formation and rejection of autologous iPSCs has been reported in mice. So there is no clear therapeutic advantage for the use of autologous stem cells. This paper provides data from monkeys that give some hope for the future autologous therapies. Let's look at the data. Authors derived iPSCs from skin tissue of 3 rhesus monkeys aged 8 to 10 years using Yamanaka factors and differentiated them into neuronal cells. Schematic of differentiation protocol is shown here. Similar to human PSCs, rhesus iPSCs differentiated to neuroepithelia that organized into neural tube-like rosettes and expressed neuroectoderm transcription factors PAX6. By day 42 of differentiation using hedgehog and fibroblast growth factor 8, 37% of the cells were beta 3 tubulin positive neurons, 16% were S100B immature astrocytes, and the remainings were largely nestin positive progenitors. No oligodendrocytes were observed at this stage, which usually appear at a much later stage during primate stem cell differentiation. The major neuronal populations were positive for GABA 49% and tyrosine hydroxylase 16%. GFP is reporter introduced into the cells for detection. So these cells are used for transplantation or grafting. Authors created a monkey model of Parkinson's disease by unilateral intracarotid artery infusion of the neurotoxin MPTP. All the 3 monkeys developed a stable hemiparkinsonian condition. Cells differentiated at day 42 contained immature neurons that were largely positive for GFP, as shown in the previous slide, were transplanted into the striatum and substantia nigra of the same monkeys 12 to 18 months post MPTP lesion without any immune suppression. Authors examined the graft 6 months post-transplantation using stereological and immunocytochemical analysis on serial coronal sections. Analysis of GFP positive cells indicated that about 63% were MAP2 positive neurons, 22% were GFAP positive neurons, and approximately 10% were myelin basic protein oligodendrocytes. The vast majority of GFP positive neurons were GABA positive and few were positive for tyrosine hydroxylase. This may explain why the authors did not observe obvious behavioral recovery. Data is not shown here. Immunostaining for Ki67 labeling mitotic cells showed no positive cells in the grafts. Again, data not shown here, indicating differentiation of grafted progenitors. No cells in the grafted brain were positive for pluripotent stem cells and for non-neuronal lineage markers. Immunostaining for CD3 and CD8 for lymphocytes on monkey brain sections showed no obvious difference between the graft and surrounding host tissues, so no immune reaction. Spleen tissues used as positive controls were immunoreactive for all the markers. Immunostaining for human leukocyte antigen D-related for microglia and macrophages revealed microglial cells throughout the brain. Immunostaining for several GFAP showed a moderate increase in intensity. These results indicate iPSCs could be used for therapeutic purposes. The scarcity of dopamine neurons in the graphs and lack of functional improvement in the animals are likely due to the neural differentiation strategy that did not yield sufficient number of midbrain neurons. Better differentiation protocol might yield better results. Let's hope for the best. Next, we will see a paper published in Science Translational Medicine. Use of antidepressant to reduce CSF A beta production in healthy subjects. Why this paper is so important? Alzheimer's disease has affected millions of people around the world and still no cure is available. Any reprieve to the patients save millions of dollars in healthcare costs and better life for patients. This paper provides encouraging data from human subjects using serotonin reuptake inhibitor Citalopram. Let's look at the data. This experiment was done in 12-month-old APP-PS1 mice. Animals were treated with various amounts of citalopram and CSF A beta was measured using microdialysis probe inserted into the hippocampus. As shown in the figure, citalopram significantly reduces A beta levels in the CSF after 21 to 24 hours after drug administration. 6-month-old APP-PS1 mice were treated with citalopram 10 mg per kg per day or vehicle intraperitoneously for 28 days and imaged using 2-photon microscopy. No new plaques formed in drug-treated mice, while new plaque formations seen in the vehicle-treated mice. So, more number of plaques in the vehicle-treated animals than the drug-treated ones. Citalopram also attenuated the growth in size of pre-existing plaques. Citalopram also reduced the appearance of new plaques but did not make disappear the plaques already present. Let's look at what happens in the humans. Human volunteers who received citalopram had lower mean CSF concentrations of A beta compared to placebo. The mean production rate of A beta over hours 13 to 22 was significantly slower in citalopram treated individuals, but the clearance rate of A beta was unchanged. These results suggest that citalopram might be useful in treating Alzheimer's patients, thereby reducing the formation of new plaques and reducing the increase in the size of already developed plaques. So, it is not a cure but might provide relief to patients and if given early on might help the patient to lead a better quality life. That's all for now. Thank you for watching Science Breakthrough from Science Mission.